Good afternoon, everyone. My name is Blake Sanchez, and I am here alongside my team, including Ryan Villacrucis and Zach Baum. Today, my team and I will be discussing the 5-HT1A receptor and its interaction with cannabidiol in the treatment of anxiety disorders. Before we jump into our discussion surrounding this topic, allow me to outline what we as a team will be discussing in today's presentation. First off, we will begin by explaining an ever-increasing issue in society, which is the prevalence of anxiety disorders and why it's a problem that needs to be addressed. Next, we will discuss how CBD interacts within the human body, and more specifically, its function within the 5-HT1A receptor. We are also going to talk about single nucleotide polymorphisms and how they can serve as potential biological markers for the onset of anxiety disorders. In addressing single nucleotide polymorphisms, we will also go over possible gene therapy that could be used. By this point, we have given enough general background to begin discussing specific firing mechanisms of 5-HT neurons. Following this, we will then discuss the importance of 5-HT1A receptors in the dorsal raphe nuclei, as well as in the dopaminergic pathway. We will then touch on the current treatments available for anxiety disorders and provide a comprehensive rundown as to why CBD should be considered as an alternative to anxiety treatments. To conclude this, we will discuss current research and the future outlook on CBD within this field. Numerous estimates suggest that nearly 7.3% of the world's population suffer from some type of anxiety disorder, making it one of the most prevalent illnesses suffered by humans. Anxiety disorders have been linked to various problematic issues such as increased rates of employment, worsened physical health, declined quality of life, increased relationship failures, and heightened suicidal risk. Unfortunately, anxiety disorders also exhibit a very low age of onset, starting in early adolescence or adulthood for many. Here on the slide on the right-hand side, you can see a graph that I have added that shows the mean age of onset for multiple types of anxiety disorders. From this, you can see that the age of onset is typically very low for most of them. That being said, anxiety disorders pose a real problem for our current and future generations, establishing the need for better treatment options in a limited array of current ones available. So many of you have probably heard of CBD, but you may be asking, what exactly is it? Well, cannabidiol, which is commonly referred to as CBD, is one of the nearly 400 chemicals existing in the cannabis sativa plant and is one of the 61 cannabinoids, which are a class of molecules that act as neurotransmitters within the brain. On the right-hand side of this slide, you can see the chemical structure of CBD. Now, recently, more and more research has analyzed CBD as a potential in treating psychological disorders such as anxiety. Now, allow me to explain how CBD interacts within the human body. Cannabinoids like CBD produce their effects by interacting with specific receptors throughout the central nervous system. CBD is essentially regulating how cells communicate amongst one another when it comes to sending, receiving, or processing messages. The vast array of receptors that CBD can bind to alludes to its possible implications in medical treatment of many diseases. However, today we will be focusing primarily on its role within the 5-HT1A receptor, which is a subtype of serotonin receptor found in our bodies. CBD acts as a nonspecific agonist in conjunction with the 5-HT1A receptor that serves to exhibit similar effects on the 5-HT system, as does the naturally occurring serotonin neurotransmitters found within our bodies. As comprehensive knowledge of the human genome continues to be developed, new light is shed on the range of unique allelic variants contained within DNA and their relationship with specific disease processes. Through the use of whole genome disorder association studies, scientists have found single nucleotide polymorphisms, also short-termed as SNPs, associated with the anxiety disorders localized in untranslated, non-coding, and regulatory regions of DNA in patients with these anxiety disorders. Now let's remember that SNPs, also known as SNPs, are the most common type of genetic variation among people, with each SNP representing a difference in a single nucleotide within a DNA sequence. On this slide, you can look here on the right-hand side and see a visual representation of what a SNP will look like. Here you have 3 identical genes except for one single nucleotide. Here you have an adenine, a guanine, and a thymine. This implies that there is a genetic predisposition to the onset of various anxiety disorders. So further research is warranted in gathering genome-wide sequencing for these SNPs in order to understand possible causative genetic mechanisms for generalized anxiety and other associated disorders. This could help scientists develop novel drugs that target specific regions of a patient's own DNA through genetic therapy treatments. One potential application of gene therapy is the correction of SNPs that have been correlated with anxiety disorder onset. The RS6295 SNP is a promoter region polymorphism in the 5H2R1A gene, the gene that encodes the 5HT1A receptor we've been talking about. The homozygous GG genotype for this allele is correlated with the upregulation of the 5HT1A autoreceptor gene, resulting in increased levels of presynaptic 5HT1A autoreceptors within the RAFE nuclei. The RAFE nuclei is a cluster of interconnected neurons deep within the brainstem containing relatively large concentrations of 5-HT1A autoreceptors. The mechanism by which this effect occurs is through the R6295 SNP, preventing the binding of the transcription factor repressors called DEF1 and NUDR, both to the silencer sequence. An increase in concentrations of 5-HT1A autoreceptor leads to lower concentrations of the 5-HT in the synapse and thus a decrease in 5-HT efficacy. A decrease in postsynaptic heteroreceptors as a result of the R6295 SNP also leads to a decrease in 5-HT efficacy at the synapse as less receptors exist to bind to the available 5-HT in order to express inhibitory downstream effects. These mechanisms are the predominant underlying causes in the development and acquisition of anxiety disorders. Further genome mapping of unique SNPs in the 5-HT system is still needed to understand more of the cause and effects. But once we know enough, more novel drugs could be created and used in gene therapy to add proper nucleotides to the mutated DNA causing the anxiety disorder.
Presynaptic 5-HT1A receptors are found in the RAFE nuclei, while the postsynaptic receptors are found in high densities in the limbic system, the system that essentially modulates memory and emotion. The neuronal firing of serotonergic neurons are homeostatically regulated by the natural ligand, serotonin, as well as a number of other agonists and antagonists. Binding of the presynaptic neurons have an inhibitory effect that decreases the neuronal firing of 5-HT neurons. The production of these neurotransmitters essentially originate from the RAFE nuclei and travels all throughout the brain. Although the overall biochemical mechanisms of CBD's interactions in the body still remain poorly understood, new studies regarding important subcortical structures of CBD's anxiolytic effects have been recently discovered. One of these structures includes the importance of the dorsal RAFE nuclei. The 5-HT1A receptors located in this region have been shown to inhibit nausea and vomiting caused by a number of diseases, including anxiety disorders. These receptors are co-localized with neurokinin receptors as well. When the activation of 5-HT1A receptors occurs by an agonist like CBD, the neuronal firing of substance P is inhibited. This substance is linked with a nausea response in the body. Substance P and other ligands competitively bind to the neurokinin receptors in this region, and if high concentrations of substance P is present, then a nausea response is induced. Conversely, if low substance P concentrations are present, no nausea response is produced. Ultimately, what this tells us is that CBD is useful and that it has the ability to inhibit the release of substance P, thus inhibiting the nausea response. To further emphasize 5-HT1A's importance for the attenuation of vomiting, CBD's ability as an anti-nausea molecule is debilitated in the presence of 5-HT1A receptor antagonists, like whey, in this region, thus emphasizing 5-HT1A's importance in the role of attenuating emesis. Additionally, the dorsal RAFE nuclei are home specifically to somatodendritic 5-HT1A autoreceptors, stimulation of which leads to the modulation of serotonin exocytosis. This leads to the suggestion that The emetic response is also linked to the presence of serotonin within this region of the brain. It was found that CBD's presence in this region was able to enhance the ability of another receptor agonist, 8-OH-DPAT, a known ligand that attenuates vomiting. This conclusion is justified in that an increased stimulation of the molecule 35S GTP gamma S was found in the brainstem. This indicates an increased number of G protein coupled receptors like 5-HT1A being activated upon CBD's agonism, ultimately telling us that the receptor agonist 8-OH-DPAT and CBD increased its rate of binding, therefore inhibiting the reuptake of serotonin and reducing the symptoms of emesis. Another important region for CBD's interactions with 5-HT1A receptors include its interactions with the postsynaptic region contained in the mesolimbic pathway. In the system, the nucleus accumbens has been implicated to be an important site for the modulatory effects of CBD. If CBD is present within the shell of the nucleus accumbens, the prevention of a conditioned fear response is observed. And this is seen through decreased locomotor activity as well as decreased episodes of freezing. Similar to 5-HT1A's role in the dorsal raphe nuclei, the presence of its receptor antagonists like NAD and A-flu shows no decrease in freezing episodes, nor a lowered activity in locomotor function. Additionally, a decrease in dopaminergic neuronal activity in the ventral tegmental area is observed when CBD is paired with the 5-HT1A receptor antagonist at the shell site. This is important because due to dopamine's role in the mesolimbic system as a modulator of anxiety, a decrease in dopaminergic neurotransmitters would mean a promoted fear response in the brain. This suggests that the inhibitory effect of CBD on the acquisition of fear expression is dependent on the activation of 5-HT1A receptors in the nucleus accumbens shell.
So current treatments for anxiety disorders heavily rely on the preliminary use of SSRIs in treating someone with these disorders. So doctors will prescribe these drugs as the first line of defense in their treatment plan for these disorders. And SSRI stands for selective serotonin reuptake inhibitors. And they were developed in the early 1970s initially, and shortly after the development of the drug fluoxetine, which was the first SSRI, the mechanism of action of those drugs was discovered. And that mechanism of action is on 5-HT transporter. And this is a membrane protein on the presynaptic neuron of the serotonergic pathway. And it's responsible for taking synaptic serotonin and via endocytosis, transporting it back into the presynaptic firing neuron. And SSRIs inhibit the function of the 5-HT transporter in order to indirectly increase the viability of synaptic serotonin. And in 2013, a new drug called vortioxetine was developed, and this is an example of the newest SSRI that is not only works through inhibition of the 5-HT transporter, but acts as a direct agonist of the postsynaptic 5-HT1A receptor to exert its anxiolytic effects. Two other drugs, so buspirone as well as the drug class benzodiazepines are prescribed for anxiety disorders. Buspirone is only a partial agonist of the 5-HT1A receptor, so it doesn't work as well as, say, serotonin does on its receptor. And benzodiazepines are more prescribed for panic attack-like symptoms that occur acutely, and they are used as such, so they're not a long-term treatment option for anxiety disorders. So increasing evidence has shown that the use of CBD is a good alternative for SSRIs, as it shows great long-term anxiolytic effects with minimal side effects. Side effects of SSRI treatment include oftentimes nausea, insomnia, panic attacks, decreased libido, and weight loss. And so these are very unwanted symptoms, and if we can avoid them with CBD, for example, and if CBD does the same action as SSRIs, why wouldn't we use it? And another reason to look at CBD as an alternative treatment for SSRIs is that SSRIs don't exactly work all of the time. In fact, they work less than 50% of the time in reaching full remission for patients. And remission is just the relief of symptoms from their anxiety disorder in this case. So current research into anxiety disorders, specifically with CBD, show the anxiolytic effects of CBD stemming from direct agonism of the postsynaptic 5-HT receptor, 5-HT1A that is, and the result of this is anxiolytic effects on the receiving neuron. Another field of research is in the genetic predispositions and underpinnings in the acquisition of these diseases. And so when we look at stuff like SNPs, stuff like genetic variants, we're learning more about the predispositions that these diseases consist of. And once we do that, as we continue to do that, we will be able to target specific symptoms better, and we'll be able to classify these diseases better and introduce new pharmacologicals that treat them better. So as CBD's role in the 5-HT1 system is further revealed, CBD's multitude of agonist sites will be also further revealed, and insights into CBD's mechanism of action on other receptors in the brain and body will relate to a myriad of fields in medicine, such as sports medicine, palliative care, and oncology. And with the development of novel treatments through the use of genetic sequencing, common variant sequencing, and analysis, we will be able to work on something called precision medicine, and that's sort of like a specialized treatment for a specific set of predispositions that a patient might have. And CBD could be involved in some of this treatment moving forward. And as we develop these better drugs for the treatment of anxiety disorders, of course, the global burden of these diseases is massive, as was pointed out earlier, and hopefully that will decrease the global burden. Thank you.